So we finished talking. Well, we started talking about how natural selection works on traits that are continuous variables. And I've already talked about stabilizing, disruptive, and directional selection. Directional selection shifts the mean value of a trait. Stabilizing keeps the mean where it is and selects against the extremes. Disruptive selection favors the extremes and selects against the mean. Let me see here. I've got a pile of your exams right here. And if anybody was curious, disruptive was actually on one of the clues in the first question, if anybody wasn't sure about that. To get a little bit more mathy, we can talk about the variance in a quantitative trait. And I don't normally make you guys calculate this, so don't worry about memorizing the formula. But the variance is the mean of the squared differences between each data point and the average. If we take the square root of that, we have the standard deviation, which is something you might be more familiar with. And intuitively, you can think of the variance as how fat your bell curve is. If variance is very low, then almost all of your values are very close to the mean and your bell curve is very narrow. If variance is high, then you have a lot more spread around the mean and your bell curve is a lot wider. And so we could take any trait in a population and measure the total phenotypic variance. We could measure, everybody's height, or I don't know, the number of leaves on every sumac plant in the nature reserve, or anything that we wanted to, and calculate the variance, and we call that V sub P. Some of the variance comes from genetic variation in the population, and some comes from environmental variation. Okay, this class varies in height. Some of you are taller and some of you are shorter. So we could calculate the total phenotypic variance in the class. Some of the variance comes from the fact that you've got different genes. Some of the variance comes from the fact that you've been exposed to different environments. You may not have been exposed to that much variance in environments because most students tend to come from a, American middle class type background. But if any of you came from a very different environment than that, then some of the variance in height will correlate with the fact that you were raised in variable environments. And we can just think of the total phenotypic variance as the sum of genetic variance and environmental variance. So up at the top left, that bell curve shows what the variance in a population would be if all members had an identical environment. Let's say we took a bunch of sumac plants and we raised them in exactly the same environment. We did what's called a common garden experiment, and we put them all in the same soil, same amount of light, same amount of water, same amount of fertilizer, made their environments as identical as was physically possible. Then all of the difference among those sumac plants would have to be due to genes because they'd be growing in identical environments. At the top right, that's the variance if all of our population members had identical genes. Let's say we cloned a bunch of sumac plants, maybe grew them from cuttings, let's say, but we raised them in different environments. Then all of the variance would be due to the environment and none would be due to genes because all of our plants would be genetically identical. So VG is the variance if all members of the population had the same environment, but different genetics. VE is the variance if all members of a population had identical genes, but different environments. And you can think of the total phenotypic variance in a real population as being the sum of the two. Sort of imagine taking those two bell curves and kind of shoving them together and you'd end up with something like the diagram below. Here's the thing, you hear about a lot of traits as being, such and such percent genes and such environment. Like, intelligence is, I don't know, 70% genes and 30% environment. Don't quote me on that. I just made up those numbers. But human height is sometimes said to be 70% genetic and 30% environmental. The big disclaimer is that only applies to variance in a population. It doesn't apply to an individual trait. When I don't slouch, I'm 6'2", but there's no way I can say that 70% of my height is due to genes and 30% is due to eating my Wheaties, right? There's no way that I can say like let me think. Oh, dang, I have to do math. There's no way that I could say, if I'm 74 inches, I can't say that 50 of those inches are due to the fact that my dad is also pretty tall, and the remaining 24 of those inches are due to the fact that my mom fed me a lot of organic food as a kid. Okay, that's meaningless. Your height is your height. Your, I don't know, number of leaves, if you're a sumac plant, is what it is. And the only time when you can start applying that, these percentages is when you're talking about the variance within a population. You could have a trait, and in fact, you often do have a trait that is, let's say, fully genetically controlled and doesn't have any variance at all. One example would be, let's take the class, the people that are hearing my voice. We know that genes have a lot to do with the development of your nose. Okay, your nose development is under genetic control when you are an embryo, when you're roughly the size of a bean, is when your nose gets built. So we know your nose is genetically controlled, but the number of noses has no variance at all. And the reason is that every one of us has exactly one, right? I'm assuming nobody who's hearing my voice has like extra noses stashed somewhere, maybe growing on some part that's covered by clothing. We're all got one nose per person, and that means the variance is 0. Why? Remember, it's the mean of the squared differences between each data point and the average. Average number of noses, 1. Each individual data point, 1, because we've all got one nose. Difference between data point and the average, 0. Square those differences, 0. Take the mean of a bunch of zeros, you get 0. So you can have a lot of traits that don't have any variance because they're all the same. Now we can take the fraction of phenotypic variance in a trait that is due to genetic variance, is called the broad sense heritability, and it's written as capital H squared. Why is it written as anything squared? Reasons. Has to do with statistics. It has to do with like math and stuff. I'm not going to worry about that. So genetic variance divided by total phenotypic variance is what we call broad sense heritability. As I was saying, you have to be really careful about this because you can have a trait that is inherited genetically and has 0 heritability if it has no genetic variance. If we had a population of clones, let's say there's 50 clones of me walking around. Oh God, that would be really surreal. Okay, there's 50 clones of me running around. Genetic variability is 0, but I actually tan passably well. So let's say we have 50 clones of me, and some of them, lock themselves in their bedrooms, and some of them go to the beach and get a pretty savage tan. We would have phenotypic variability in skin tone because some of these clones would be pale and some would be uh, fairly dark, but we'd have no broad sense heritability because there would be no genetic variance because clones aren't different from each other. That's a little weird thinking about clones of me, but you'll see an example of this sort of thing coming up, not done on humans. And as I was saying, you could have an undefinable heritability if a trait doesn't have any variance at all. So if I tried to calculate the broad sense heritability of the number of noses per person, I'd get 0 divided by 0, right? I would not be able to calculate heritability. So heritability is not the same thing as being genetic. You can have lots of traits that are genetic in the sense that genes are responsible for making you have them, but that either have no heritability or have undefined heritability. You can go a little farther with this and define something called the narrow sense heritability of a trait, and that's written lowercase h squared, and that is equal to the additive genetic variance divided by the total phenotypic variance. Oh God, what's that? You can divide genetic variance into V sub A, V sub D, and V sub I. That's all down here. V sub G is the fraction of variance in a population that's due to genetic variance. V sub A is genetic variance due to additive effects. V sub D is genetic variance due to dominance. And V sub I is genetic variance due to interactions. What the hell is that? Let me show you an example that hopefully will make this a bit clearer. And I'm actually going to go to the whiteboard in just a minute. Here is the scarlet tiger moth. Used to be called Panaxia. It's now in genus Callimorpha. And it turns out that Panaxia has spots on the forewing, and their number is governed by a single gene with 2 incompletely dominant alleles. Down here on the bottom, this one is homozygous recessive, and you can see this one only has 2 white spots on each forewing. Up here, the heterozygote has 4 big spots on each forewing, right there. And up here, the homozygous dominant, phenotype typica, you can see it's a lot more spotty. By the power vested in me by the state of Arkansas, for reasons that'll make this a little bit more clear, I'm going to say that has 1, 2, 3, 4, 5, 6 spots on the wing. Now, yes, there's a few more that are out there, and I'm not calling them spots, but that's because there's a point I want to make. So we have 2 spots on the wings of homozygous recessives, 4 on the wings of homozygous heterozygotes, and 6 on the wings of homozygous dominants. Now, what can I do with this? Okay, so we've got tiger moths. If you have genotype SS, you have 6 spots. If you have genotype big S little s, you have 4 spots. And if you have genotype little s, you have 2 spots. All right, what am I going to do with that? Well, I'm going to draw pair of axes. There's an axis. There's an axis. And I'm going to graph mean parental phenotype against mean offspring phenotype. All right. Okay, close enough. So how are we going to do this? All right. Let's say we have two SS parents, or two little s parents, right? One parent has 2 spots per wing. The other parent has 2 spots per wing. The average of 2 and 2 is 2. So the average parental number of spots per wing is 2. All of the offspring are also going to have 2 spots per wing. So we can put a dot right there. If we cross two little s parents, their mean phenotype is 2 spots, and all of the offspring will also have 2 spots each. So the mean parental phenotype is 2, and the mean offspring phenotype is also 2. If we cross two big S moths, both parents have 6 spots, and all of the offspring will have 6 spots. So the mean parental phenotype is 6, and the mean offspring phenotype is also 6. Okay, what if we cross a big S tiger moth with a little s tiger moth? Well, one parent has 6, one has 2, and the mean is 4. All of the offspring will be heterozygotes, and they will have 4. So we can put a spot right there. Let's say we cross a big S and a big S. One parent has 6, one parent has 4. So the mean is 5. And half the offspring will be homozygous dominant and half will be heterozygous. So the offspring mean is also going to be 5. So we can put a spot right there. If we cross big S with little s, right, one parent has 4 and one has 2. The mean is 3. Half the offspring have 4 and half the offspring have 2. So the mean is also 3. If we cross two heterozygotes, the parent mean is 4. And then a quarter of the offspring have 6, half have 4, and the other quarter have 2. And the mean of all of that is going to be 4 as well. So you see what I'm getting at here. Hopefully what you've noticed is that all of these points lie along a line with a slope of 1. This is an example where genetic variance is entirely additive. The only thing that is making tiger moths different from each other is the fact that they have different numbers of alleles. If you have no big S alleles, you have 2 spots. If you've got 1, you have 4 spots. If you have 2, you have 6 spots. That's a case where all genetic variance is additive genetic variance. Okay, that's an easy example of additive genetic variance. Now, if you try this with a different species, if there is dominance going on, let's say for a moment, let me go back to the slides. If we pretend for a moment that the number of spots is governed by a single gene with two alleles, one is completely dominant to the other, then not all of your variance is due to the additive effects of genes. Some of it's due to dominance. Some of what is making these moths different from each other would be genetic dominance. In the real world, everything that's making these moths different about each other, at least as far as wing spotting goes, everything that makes them have different numbers of wing spots is the number of big S alleles that they've got. So their genetic variance is not only, or their variance is not only genetic variance, it's entirely additive. But in other species, in other cases, some of what makes your critters genetically different from each other is the effect of gene dominance. And some of what makes your critters different from each other is the effect of gene-gene interactions. The reason why my cat is gray tabby has to do with interactions between one gene that governs the presence of light hairs with dark tips and a second gene with different alleles that govern where those go. So part of what makes all of my cats different from each other is not just that the fact that they've got different genes, but the fact that the genes are interacting with each other in different ways. So that's the difference between additive genetic variance, which is due purely to the number of copies of alleles that you've got, genetic variance due to dominance, and genetic variance due to interactions among genes. The tiger moth is an example where, at least as far as wing spotting goes, all of the variance is additive genetic variance. But in many cases, this is not what you find. And notice something. Let me go back to the whiteboard for a second. Notice that in this case that we just did, when you have nothing but additive genetic variance, all of these points ended up lining up on a line. And that line has a slope of 1, right? It's just y equals x. That's got a slope of 1. You can estimate narrow sense heritability as the slope of this line. If you have a population, you can calculate the mean parental phenotype and the mean offspring phenotype, plot them like this, and your estimate of the narrow sense heritability is the slope of the best fit regression line. Here it's 1. If we had genetic dominance going on, the slope would not be 1. It would turn out to be a bit less than 1. In fact, while I'm thinking about it, I need to remember to email this out to everybody, but while I'm thinking about it, we might as well do that now. Every time I've taught this course since 2012, I've asked students voluntarily to find out the average of their parents' height and the average of they and their full siblings' height. I never require people to do that. I certainly don't ask for more information than height. And I never require anybody to do that. Some of you might be adopted or have other interesting family situations, and that is entirely your business and not mine. So you don't have to do this if you don't want to for any reason. But for those who can, if you can report to me the mean of your parents' height and the mean of the height of you and your full siblings, we can use that to calculate an estimate for the narrow sense heritability of human height. And in fact, I've got a data set that I've been gathering for, again, I think since 2012. And yeah, we've actually got a pretty good estimate of the narrow sense heritability of human height. So I'm going to ask for that on Wednesday. And again, if you don't want to participate, no problem, no harm, no foul. You know, it's all good. You won't get penalized. But if you can add just a few more data points as we try to estimate human height heritability. Okay, great. Let's go back to slides. The reason you might actually want to know narrow sense heritability is that it feeds into one equation that I am going to ask you to keep in mind. And this is called the breeder's equation. And it's very simple, R equals H squared S. H squared, that is narrow sense heritability. That's what we've done. R is the response to selection. That's the difference between the mean trait in the parents and the mean trait in the offspring after one round of selection. Okay, after one generation, the difference between mean trait in parents and the mean trait in offspring after one generation of selection is the response to selection. And then S, whoops, sorry. S is called the selection differential. That's the difference between the mean in the general population and the mean in the population that you've selected for breeding. Okay, let me go back to the whiteboard and try to diagram this. Okay, there is our whiteboard. I'm going to clear this if that's all right. And let's clear it. Okay, let's say I am, I don't know, let's say I'm a chicken farmer. Okay, this is Arkansas. The state bird is the Tyson chicken. And what I want to do is raise chickens that are super meaty, lots of meat on their bones, because that's the way I'm going to make my money. All right. So what we have here is a bell curve. Right here we have, let's just say it's weight of the weight of our chickens. And our distribution is going to be some sort of bell curve, because we have, you know, some chickens that are lightweight, and most of them are going to be kind of average. And then some of them are going to be humongous. All right. So if this is a true bell curve, then the curve should be symmetrical. And right up here, we should have the mean. And I can represent the mean weight as W bar. We've seen this before, where we use bars to show mean fitnesses, but a bar over a number in general just means it's a mean. And so if this is a true bell curve, if it's not skewed, then right up here we have W bar. Okay, let's say I want to breed meatier chickens. Well, I think to myself, okay, I'm going to take a cutoff point. The point of selective breeding is you pick out the individuals that you want to breed and get rid of the ones you don't. So I'm going to assign a cutoff weight. And let's say that the cutoff weight is right there. A chicken that is above that cutoff weight will be allowed to breed. A chicken that is below that cutoff weight, you know, all of the chickens in here kind of trying to X out that zone. All of the chickens that are in here are going to end up in the stew or in the fricassee or whatever it is that you do with excess chickens that you no longer care to keep around. So all of these chickens get it right in the neck. All right, all of these chickens go off to the big Tyson plant in the sky and are never seen again. Only the chickens here are the ones that get to reproduce. All right. Well, this population of chickens right here, they have some mean, right? The chickens that I'm allowing to live, I can calculate their mean weight, and I can call that W bar star. There's that bar. W bar star. That is the mean weight of the chickens that I allow to live. All right. And my selection differential, which I'll write capital S, is equal to, this is going to take me a second because I have to draw some characters in. All right. The selection differential is going to be equal to the absolute value of W bar minus W bar star. Okay, you're with me there. To calculate the selection differential, I take the average trait value in the population before selecting, and then I do my selection and decide which chickens will live and which ones will breed and which ones will get shipped right off to Springdale and turned into consumer products. And I take the absolute value of the different trait averages between the population before selection, which is W bar, and the population after selection, which is W bar star. Okay, let me see if I got that right. I calculate the mean of the trait before selection, and then I do my selection, and I calculate the mean of the trait after I've done selection, and I take the absolute value of the difference between those means, and that is the selection differential. It's a measurement of how strict my selection is. If I were to allow, all of my chickens to breed except for the very tiniest, if I were to allow all of my chickens to reproduce except for these, then my selection differential would be very low. On the other hand, if I was very strict about this and only allowed the absolutely biggest chickens to breed and every other chicken, all of these ended up as, whoops, let me erase that. That wasn't what I want to do. Okay. There. All right. If I wiped out all of my chickens except for the absolute elite way out here at the very far end of my distribution, I would have a very large selection differential. The difference between the average before selection and the average after selection would be very great. So how big S is a measurement of how stringent my selection process actually is. Okay, what's R? Let's go back to where we were. And let's say that I have oft most of my chickens, all of the chickens that are below this height, get shipped off to Mr. Tyson and turned into McNuggets. And all of the chickens that are above this height are allowed to breed. Okay, so I take these chickens, I let them breed, and once the offspring have fully grown, that's a good color. Once the offspring are full grown, they will have some they will have some weight distribution. And if I graph the weight of the offspring after one round of selection, I might end up with something like this. Okay, that's a pretty shoddy bell curve, but what the heck? Right? Some of my offspring are going to be small. Some of my offspring are going to be huge. Most of my offspring are going to be somewhere in the middle. And if it's a normally distributed bell curve, then I will have, some mean right here. There'll be a mean size, a mean weight of my chickens, which I can represent as X bar. All right. The response to selection is going to be equal to the absolute value of the difference between I've got to draw a couple of symbols there. It's going to be the absolute value of the difference between these two means. If that difference is very low, then selection didn't do very much. The population is still much like it was when we left it. If that difference is very large, that means that selection has really shifted that mean over. So S is a measurement of how stringent my selection is. R is a measurement of how much response we get over one generation. And the breeder's equation is that S is equal to narrow sense heritability. Whoops, got that backwards. The response to selection is equal to narrow sense heritability times the strength of selection. The response is equal to narrow sense heritability times this is also called the selection differential. So there was supposedly a turkey farmer in the 19th century in America, a guy named Ignaz von Rahl, who got the brilliant idea of putting turbans on the heads of his turkeys. Supposedly this was because he thought he could breed a new variety of turkey in which the babies would hatch with turbans already on their heads. This equation shows you mathematically why that simply isn't going to work. I mean, like you couldn't already tell. This is kind of an extreme example, but you can already tell why that's going to work. And the reason it's not going to work is that wearing a turban on your head has a narrow sense heritability of 0 because it has nothing to do with your genes. There are no genes that make you wear a turban. Not if you're a human and not if you're a turkey. If you're a turkey, you wear a turban because the stupid farmer put one on your head, but you can't be born with a turban. So if narrow sense heritability is 0, then you can select all you want to for wearing turbans on your head, and your turkeys will never develop it. The response to selection is always going to be 0 if the narrow sense heritability is equal to 0. More generally, if narrow sense heritability is low, what that means is that artificial selection is not going to produce any particularly big response. You can select and select and select, and if narrow sense heritability is low, the response is going to be very small. On the other hand, if narrow sense heritability is large, if it's close to 1, then very stringent selection will give you a very big response. So if you can calculate narrow sense heritability, you can predict how well a selective breeding attempt is likely to work. That's what the point of all this is. This kind of went around for a bit, but hopefully you get the picture. So we'll go back to the slides just for a couple of minutes, and then I'll give you clowns a break. So that's the breeder's equation. And as I was saying, this is some old data. This is class data for spring of 2012 and summer 2013. The current data set looks a lot bigger than this because I've been gathering this data every time I've taught this course. But I asked everybody to bring in their mean parental height in inches. That's on the x-axis. And the mean offspring height on the y-axis. That is the height of each student and all of their full siblings averaged out. And when I plotted that, what I got was a line with a slope of 0.915. I don't know if you can see it, but that's the regression equation right there. The accepted value is 0.7, and I think we're closer to 0.7 because I've added more data from every evolution class I've taught since the summer of 2013. And with any luck, you can be next. But yeah, the accepted value of narrow sense heritability of human height is 0.7. This estimate came in a little bit high, but with adding more data, I think we've actually gotten quite a bit closer. So this is how you can estimate narrow sense heritability and then plug that into the breeder's equation, which is very useful if you're doing artificial selection. You can do it in studies of natural selection as well. Here you've got on the x-axis, you've got mid-parent bill depth, right? That's the average of the bill depth of both parents in a population of the medium-beaked groundfinch, Geospiza fortis, on the island of Daphne Major. And you can see in 1976, you have this very nice linear relationship between mid-parent bill depth and mean offspring bill depth, right there, all of those dark dots. In 1978, that was after a big drought, and drought exerted selection because drought drastically affects the seeds that were available. And in 1978, in 1978, your regression line is down there. So there's been selection going on, but the slope of the line hasn't changed. And that strongly suggests that we are measuring something that's genuinely got to do with genes, because a year of very strict selection moves the line, but doesn't change the slope at all. So it looks like beak size in these Galapagos finches is under pretty stringent genetic control. And the fact that the line shifts means that because this trait is under strong genetic control, it has a high narrow sense heritability, and selection can affect the trait quite a bit. Beak size responds to natural selection because that's what it did when the drought hit. This is actually a pretty good place for me to take a pause. So I'm going to go ahead and stop the recording.